Welcome to Biomentors. Today we are going to discuss a topic from Plant Tissue Culture lecture series. We are going to discuss about Meristem cultures. So let's get started. So what is a meristem? So meristem means it is a part of plant which plays a key function to increase the plant length. It refers to the meristematic cells. So meristem is a group of cells which are termed as meristematic cells, and this meristematic cells functions to increase the plant length because these cells are actively dividing or continuously dividing cells. There is no intercellular space between the meristems. Now, what is meristem culture? Meristem culture can be defined as a technique in plant tissue culture which makes the use of apical meristems. So there are three types of meristems generally seen in plant. First is apical, which is at the top portion. Then there is intercalary meristem, and there are lateral meristems. So in the plant tissue culture, in the techniques of plant tissue culture, we use apical meristems, and from this apical portion is taken of the plant, and with the help of apical meristem of the plant, uh, we can get newly developed plantlets by vegetative propagation. Now the meristem can can be cultured by isolating the stem. So suppose this is the stem of the plant and this is the meristem. So we have to cut this meristem in V-shaped cut. V-shaped cut is done, and then we have to take this meristem. In this technique, by culturing the shoot meristem, adventitious roots are generated. So as we are taking the shoot meristem, we will develop adventitious root formation. The size of the shoot generally taken in the meristem culture is 10 mm, and with the help of meristem culture we can generate virus-free plants. So this is the main advantage of meristem culture. We can produce virus-free plants out of it. Now the discovery of meristem culture. It was discovered in 1952 by Morel and Martin. These were the first scientists to develop the technique of meristem culture from in vivo virus eradication of Dahlia. So Dahlia was the first plant on which the meristem culture was done by Morel and Martin in 1952. And then after, with the help of micropropagation, Morel in 1965 used this technique in orchids, on orchids. Now virus-free culture. So we can develop, as we know, we can develop from the meristem culture, we can develop virus-free plants. So what are this meristem? Meristem is the shoot tip, as we discussed, or the apical region of the shoot. And this shoot tip is generally dome-shaped, and it is the thickness of the best, we can get best result by using 0.1 mm diameter of shoot and 0.25 to 0.3 mm length of the shoot. This is the best size of the meristem culture taken. The apical shoot meristem is a portion where a stem elongates, and it was discovered in 1949 that this portion was pathogen free, proved that they are not harmed by pathogens. Why? Because as they are actively dividing cells, so pathogen cannot reach to this part of plant. And it was discovered by Limset and Cornet. It was discovered, sorry for my pronunciation. And these two scientists discovered that the apical portion of the plants are actively dividing, and free from pathogens. So for this reason, Meristem culture is very popular among the techniques in the production of virus and other related organisms free plants. Now the process of meristem culture. So first, the process of meristem culture involves the following steps. So we will see what are the steps for the production or the process of meristem culture. So first we have to select a plant and we have to take that twig of that plant, twig of the healthy plant, and then they are surface sterilized. Generally it is surface sterilized with sodium hypochlorite 1% for 10 minutes, and then after this uh, this twig is rinsed with distilled water and they are then transferred to LAF for the inoculation. So they transfer eggs plant into the petri dish, and then after removal of the outer leaves are done, and the apex is exposed. And so now we can see the apical meristem of the stem, and we have to carefully cut this apical meristem. By the method, we have to cut this apical meristem in V shape, right? And then after cutting the meristem, we can inoculate this meristem on a petri plate, right? We have to inoculate this meristem on a petri plate, on a nutrient media. And then after incubate this meristem for 16 hours of light and 25 degrees Celsius for about 20 to 25 days. And after some time, they will develop long shoots without roots. When this stage is seen in the plant, they are quickly transformed to hormone-free medium to develop roots. And then once the root is formed, the multiplication stage occurs. Then after this, plantlets are multiplied. And the last stage, which is hardening and field trial, is done. And then finally we get virus-free meristem culture, virus-free plantlet from meristem culture. So this is the diagram showing the overview of meristem culture, in which we have to take first meristem culture, meristems of the plant. The shoot tip is taken. Then after they are surface sterilized. Then they are transferred onto the medium, inoculation. Then incubation step. Generally fluorescent light is used for photo for the light, and the light photo period is 16 hours and temperature is 25 degrees Celsius. Then multiplication of this meristems is done, and finally the last stage is hardening of the plant. No, generally MS media is used for meristem culture because it has concentration low concentration of salt for majority of the species. So for the majority of the species, MS media, Moraceae and Skog media is used in meristem culture. Fungicides and antibiotics such as Meristem and streptomycin is added to remove endophytic contamination. So to prevent contamination, fungicides is also added. Factors affecting the meristem culture. There are two factors which affects the meristem culture. The first is the age of the plant or age of the tissue. Development is achieved by using young tissues as they are actively growing to assure the best chance for the success. So we have to take young tissues or young cells of the plant. And second is true success will be achieved only on early attempts, because at later stage it can cause damage to the tissues. As these meristems are very fragile, we have to culture this meristem very fast. We cannot store this meristems for a longer period of time. And if we store this meristem, then it will cause damage to the tissues and we cannot get a proper result. Now, we have seen the overview of Meristem culture. Now we will look upon the advantages and disadvantages of Meristem culture. So the first advantage of the Meristem culture is virus-free production of plantlets. The second advantage is that we can store the germplasm of seeds and plant and conserve it in vitro, or by cryopreservation methods. The the third is that meristem contains high concentration of oxygen, and thus the growth of the meristem culture is also very fast compared to different cultures. It is very effective method in the field of agriculture and industrial sciences, because virus-free plant and disease-resistant varieties can be obtained with the help of meristem culture. The example of the meristem culture which is used on the plants, the experiments, is the tobacco mosaic virus, TMV virus, has been eliminated from Petuna species. Petuna is a flowering plant, ornamental flowering plant, and it was affected by tobacco mosaic virus, TMVs, and with the help of meristem culture, this TMV virus can be prevented, and thus we can get virus-free plants. The second example is of cauliflower, brassica, in which the brassica olerus was affected by cauliflower mosaic virus, CMV, and this virus is also eliminated with the help of meristem culture. And now disadvantages. There are very less disadvantage of meristem culture. The only disadvantage is that the isolation of meristem culture, meristems are quite difficult, and we have to take young tissues to culture. Explant take more regeneration time to grow. Sometimes for few species, this disadvantage is there, that eggplant takes very long period to grow. But the major disadvantage is that during isolation the meristem could get an injury, and it is then difficult to produce new plantlets. So this was all about meristem culture. For more such videos, do subscribe to our channel, and thanks for watching.
Thanks for watching and do subscribe to our channel.